All right, looks like everybody's completed the quiz here. All right, so today we're going to talk about photosynthesis and gas exchange. Right? I'm going to warn you ahead of time, it is a lot of content. All right? I'm going to do my best to go slowly through this. I know I have a habit of talking fast and moving fast. I will do my best to slow down. It's also being recorded, so if you do miss anything, there is that recording up there. And just as a reminder, as you take notes, right, try to focus on what I'm saying rather than everything that's written on the slide, because there's a lot of words on the slides today. All right, so quickly, plan for the day though. We're going to go through this slide. There is a break built into these slides today. And then we're going to do a quick little assignment, which I think is kind of a fun assignment. And then we're going to tear down your gravity lab, and I'll talk just a little bit about the report that is due. Right? I am almost done grading your Lab 2 lab reports, so I will get you feedback on that like early afternoon. I have like 3 of them left, and then I'll upload all the grades at once and give you all feedback. All right, so let's talk about photosynthesis. So I already showed you this slide here, right? Photosynthesis is broken down into 2 reactions, and we're going to dive deep into each of those reactions today, right? We're going to start by looking at the light reactions. So just to reorient you to where these reactions are happening, right? We are in that chloroplast, so that organelle inside of a cell, and the light reactions specifically are happening in the thylakoids, right? In this reaction, we're using the energy from the sun to make oxygen and these 2 other molecules, ATP, which we have talked about, and NADPH, which I will elaborate on. And in the meantime, right, oh, just kidding, but we're also going to use water, right? Water is going to be a major component that is used here, and we will see how water is used. All right, so overview of the light reactions. I mentioned we're going to create a couple of different molecules, right? Oxygen is going to be created as a byproduct here, right? NADPH is another molecule, as well as ATP. So ATP, remember, that's kind of like an energy currency of the cell. It can transport and store energy, right? We talked about how ATP, its energy is stored in these phosphate groups. Right? When it loses or one of those bonds breaks apart in the phosphate groups, it releases energy that can power different chemical reactions. Right? But we're also going to be making NADPH. Right? So NADPH is an electron carrier. Right? It can carry an electron to where it needs to be, essentially. Right? So NADPH, right? It carries that electron, and it often carries an electron that will provide energy for cellular reactions. So it's providing energy in a different way than ATP is. ATP, the energy is stored in those phosphate groups. You have to break apart some bonds. NADPH, that energy is essentially in this electron that is being carried. Right? I saw this meme a while back, and I thought, oh, I had to include it. Right, because it's such a great visual, right? So Sam can carry the electrons, right? But he can't carry the energy for you, right? But the energy is in those electrons. All right, we will see how NADPH plays a big role in the light reactions, but I just wanted to mention, what it is and what it does. All right, light reactions. There's a lot going on, right? We're going to break this down step by step, right? And as we go, if I am going too fast, if you have questions, just raise your hand. I will not be offended. Let me know if I need to repeat something here, right? The light reactions, like I said, are happening in those thylakoids, right? Inside of the chloroplast, specifically in the thylakoid membranes. So we can see our membrane here. This is our phospholipid bilayer. Has those phospholipids, right? The tails are oriented inwards. It's this double layer. That is our membrane of our thylakoids. Right? And we're going to be utilizing a bunch of different proteins that are embedded within the thylakoid membrane here. Right? Now, as we go through and talk about all these steps, I want you to pay attention to the movement of the electrons, right? Remember, when the light is absorbed by these pigments, it excites those electrons, right? That electron that's been excited is going to be moving throughout these different proteins throughout the light reactions here. All right, so let's break this down. All right, the light reactions. Two of the major proteins that are involved are called photosystems, right? Photosystems contain those complexes of pigments that we talked about, right? That antenna complex contains 200, 300 chlorophyll pigments, right? Some accessory pigments in there. They contain those complexes of pigments. They also contain that reaction center where that electron will eventually be donated, right? So there are two photosystems, and they are named photosystem 1 and photosystem 2, right? They are named in the order that they were discovered, not in the order that the electron will use them, all right? So even though photosystem 1 was discovered first, we will see that this is going to be the second of the photosystems that will be utilized, right? So the electron actually goes through photosystem 2 and then photosystem 1, all right? So don't let those names mess you up when you think about the order of things here, right? So like I said, in these photosystems is where that light is actually excited, right? So again, I'm going to reiterate. I'm going to do a lot of reiterating in these slides, right? Yes?
Sorry, Rena, but is the lecture being recorded?
It is being recorded, yes. Thank you for reminding me, too, because sometimes I do forget, and this is one that you will probably want to go back and watch. Yes, so thank you for reminding me. Which also reminds me, I should put on captions as well. I always forget to do captions. All right, so reaction centers, antenna complexes, they are part of these photosystems, right? Both photosystem 1 and photosystem 2 will contain these two parts. They will contain that antenna complex that we talked about yesterday, right? The 200, 300 different pigments all clumped together into that antenna complex, right? That's where we're directly absorbing that light, right? That electron is being excited, right? That electron will bounce from pigment to pigment to pigment until it ends up in this reaction center, right? So the reaction center is the second bit of these photosystems, right? And this is where the electron is going to be transferred to a molecule that can capture it, right? We talked about once that electron is excited, it can either release that excitement via heat, via light, or being captured, right? In this case, it's being captured by another molecule. Now, there's some numbers associated with the reaction center here, and that is the only difference between our two photosystems, right? Photosystem 2, the reaction center is called P680, right? It has to deal with a specific wavelength that it will end up actually absorbing, right? We're not going to get into the specifics of how it gets to that wavelength, right? When it's excited, you would put an asterisk behind it, which is why you see this P680 asterisk, right? Photosystem 1, instead of P680, has P700. When it gets excited, we would note it as P700 asterisk, right? Now, when it has that asterisk behind it, right, it's excited. It has that excited electron that's been carried to it, right? And that energy with that electron, again, is unstable, so we quickly have to pass that electron off to that molecule that will capture it, and that is what's happening in this reaction center, right? That electron is coming into this reaction center. Reaction center is now excited. It's going to pass that electron on to a molecule that can actually capture it. All right. Let's start walking through the diagram now. All right. So, like I said, a lot going on here, right? But the first step is that this electron is going to be excited in photosystem 2. All right. So, light is going to excite that electron, right? That electron that is excited is very unstable, right? It's going to bounce from pigment to pigment to pigment, end up in that reaction center that will give it away to a molecule, right? That is what is happening in this photosystem 2 here, right? So that excited electron, right, is being captured, given to a molecule, which can then go through, right, a series of steps here called the electron transport chain, right? So what's happening in the electron transport chain is simply that. The electron is being transported along, right? It's being given away to a bunch of different molecules here. All right? So photosystem 2 into the electron transport chain. Right? Again, photosystem 2, right? Big first step here. I'm really elaborating on these photosystems since they play such a huge role, right? We have that light coming in, right? It's exciting an electron on one of those pigments, right? That electron is going to bounce from pigment to pigment to pigment until it ends up in the reaction center, right? This reaction center is P680 with that asterisk, meaning it's excited now, right? That electron will be given away to a molecule that can accept it, right? And this is where water comes into play, right? This photosystem has now given away an electron. It has to get an electron back, right? It's missing an electron because it gave it away. We have to get an electron, and we get that electron from water, right? So photosystem 2 is really incredible in that it's one of the few protein complexes that can actually split apart a molecule of water. And so we see that doing it in this last step here. We have water coming in, our H2O, right? It's splitting apart and giving away an electron to replace that electron that was missing. Right? And because water is splitting, we're getting some oxygen out of it. Right? So this is where oxygen as a byproduct comes into play. All right? So again, emphasizing that this is a really unique protein, right? It's one of the few protein complexes that can break apart water in this way, right? So we're breaking apart water, replacing that electron that had been excited and was shipped off from that photosystem 2. We're replacing it with an electron from the water, right? Now, eventually, in this process, and we will come back to this too, right? Because water is being split, right, it's giving away that electron, it's producing oxygen, right, but the other half of that water is these hydrogen ions, right? These hydrogen ions will come into play when we talk about how ATP will be made, right? So, splitting it apart, giving away an electron, producing oxygen, generating some hydrogen ions. All right, a lot happening in those photosystems, right? But like I said, once that electron has been captured, right, it's going to go through a series of steps in the electron transport chain, right? This is going to contain a series of molecules that will kind of transport that electron along, right? And I think for this class, there is absolutely no need for us to learn every single molecule that it is going to go through. I just want you to recognize that this electron is being transported along and utilizing a bunch of molecules along the way. Right? As this electron is moving, right, through this electron transport chain with these series of molecules, right, that movement is actually providing some energy for these hydrogen ions to go into our thylakoids, right? Again, this is going to be helpful when we're producing ATP, and we'll come back to it. So not only is this electron simply moving, right, it's also drawing some hydrogen ions into the space in our thylakoids. All right, when it's done with the electron transport chain, that electron will be given to photosystem 1. Right? Photosystem 1 acts in very much the same way as photosystem 2. Right? By the time the electron reaches this photosystem, right, it's lost a lot of energy. Right? Just the simple movement of that electron through all those molecules, right, it's lost a lot of that initial energy that it first absorbed from the sunlight. So photosystem 1 is going to re-energize that same electron, right? So our electron is coming into photosystem 1, right? It's lost a lot of energy. Light is going to hit this photosystem 1 in the same manner that it hits photosystem 2, right? It's going to excite that electron that was given to photosystem 1, right? That excited electron will once again end up in the reaction center, become excited, and be given away to another molecule, right? Same series of steps. The only difference here is that we don't need water, right? We don't need water because there's no electron to replace, right? The electron came from this initial step, traveled through those series of molecules, ended up in photosystem 1, right? So essentially photosystem 1 gained its electron from photosystem 2, right? So it has an excess of electrons, which means we don't have to replace an electron, so we don't have to split apart water, right? So that's a big difference here. We don't need to split apart water in this photosystem 1. All right, so our electron has now become reinvigorated with that light energy. It is now once again excited, right? It's going to go through yet another series of molecules in yet another electron transport chain, right? So it's becoming excited. It's going to be passed from molecule to molecule, right? Eventually, right, that electron is going to combine with a molecule called NADP+, right? It's going to bring in a hydrogen ion, and we're going to produce NADPH, right? Which we talked about in the beginning, right? It's this molecule that carries an electron, right? And the process provides a form of energy, right? So that is the main product of the light reactions here is NADPH. Right? Now, in the meantime, we also saw oxygen is being produced, right? It's a byproduct of water splitting, right? And we're also going to talk about how ATP is also being produced, right? So that electron is going from photosystem 2, being excited, to a higher energy state, losing some of that energy in the electron transport chain, becoming re-excited in photosystem 1, losing some of that energy through another electron transport chain, before finally being captured in the molecule NADPH, right? That is the final electron acceptor, right? Our electron is not going to move any further once it reaches NADPH. All right, I like to think of this as a Z scheme, right? If you think about it in terms of the energy associated with this electron, right? If we look at the amount of energy here on our vertical axis here, right? We see that we have our electron. It's being excited, lots of energy, right? Going through that first electron transport chain, losing energy, becoming re-excited in photosystem 1, and then losing a lot of energy before reaching NADPH, right? So this slide is just kind of an overview of what is happening here. But I think it's a great visual as you're thinking about the light reactions. You can think about it as the energy being excited in kind of the Z format or zigzag format. Any questions so far? It's a lot, and I will give you a chance to digest it. All right, there's one more thing we have to talk about, and that is creating ATP, right? ATP is this major molecule, right, that carries energy and provides energy for these chemical reactions, right? In our light reactions, we're producing ATP, right? So we have the whole reaction system we just talked about in our thylakoid membrane here, right? And we talked about these hydrogen ions, right? We're going to focus in on these hydrogen ions now, right? Because they play a key role here, right? So essentially, throughout the light reactions, we're creating a gradient, right? Where there's a lot of hydrogen ions on the inside of our thylakoid versus the outside of our thylakoid, right? So on the inside of our thylakoids, we have an abundance of hydrogen ions, right? Versus the outside, much fewer. So we're creating this gradient, right? Now, we like to form an equilibrium around our membranes where it's equal on either side. Right now, we do not have an equal amount of hydrogen ions on either side. There's more on the inside, right? So the hydrogen ions need to leave the thylakoid, right? And the hydrogen ions are going to leave this thylakoid using, right, this molecule called ATP synthase, right? Essentially what's going to happen are these hydrogen ions are going to be funneled into this protein. As it's being funneled into this protein kind of, it spins, it does some really cool things, right? And it combines a phosphate group with ADP, and the result is ATP, right? So the movement of those hydrogen ions through the molecule ATP synthase is generating ATP, right? And that's major, right? We want that ATP. And we will see how we use that ATP in the Kelvin cycle in the dark reactions, right? So we had generated this gradient, and we did it in 3 different ways, right? We already talked about all these ways, right? But let's go over them again because they might not have been obvious because we weren't focusing on that gradient being built, right? So now with the focus of building up this gradient, right, we are creating these hydrogen ions on the inside of our thylakoids by that splitting of water, right? That splitting of water is replacing that electron. It's producing oxygen, right? It's giving us some hydrogen ions, right? So that's one way. The second way is when we move that electron through that first electron transport chain, right? So it's the movement between our 2 photosystems, this electron transport chain. It pumps some hydrogen ions into the middle of our thylakoid, right? That's the second way we're setting up that gradient. And the third and final way is the formation of NADPH, right? When that electron is accepted by NADPH, right, we're combining 2 different things. We're combining NADP plus and a hydrogen ion with that electron to create NADPH, right? So we're taking a hydrogen ion and also kind of capturing it into this end molecule, which reduces the number of hydrogen ions on the outside of our thylakoid, right? Those are 3 ways that we're making this gradient. All right, how are we doing? It's a lot. I know I'm combining like a bunch of different concepts in biology into one lecture here. I've done my best to simplify it. All right. So overview of the light reactions. Right, we're creating 3 things. We're creating oxygen, right, by the splitting of water. Right? We're producing NADPH. Right? That's the molecule that accepts that electron in the end. That electron's not going to go any further than NADPH. Right? We're also producing ATP by using that gradient built up of those hydrogen ions. Right? So those are our 3 products. All right, we will see how NADPH and ATP will then be further used by our chloroplast, right? But before we do so, stop working, right? Would that affect the production of oxygen? Right? I'll give you a couple minutes to think about it.
Curve is like you need to meet up and it's like you can't allow it to.
Okay,
Hey Raina, I got a question.
Yeah.
So I forgot to just circle back, so I lost that there. then I got the 1 there. But you said we're getting that back.
Yes, which is that plus 1. Oh, hold on. I miscalculated yours. I read it as 2.5. Mind if I take it back?
Yeah, for sure.
Yes. Thank you. See, this is why I have you check it over, because I can miss those little things.
Sweet. Thank you.
So we can split
Yeah.
overall policy.
But thank you.
Thanks for pulling it out. All right, what are we thinking? All right, there's a mutation in photosystem 1. What is that going to do to the production of oxygen?
I feel like there wouldn't be really a whole lot of effect on the oxygen production because that is reliant more on the water uptake of the plant, rather than or water uptake of the plant and the use of photosystem 2 rather than photosystem 1. But what it will possibly do is decrease the amount of ATP that's going to be produced in photosystem 1 after the use of photosystem 2 giving an electron. So that would create a difference in the gradient from the inside to the outside of the cell. So rather than having electron ions going out of the cell through an active transport, it would have to come, or through a passive transport, it would have to come into the cell through active transport using more energy.
Yes. So let's break down what you said into different pieces so that we can all maybe kind of regroup and talk about it. So we are splitting apart water, right, to get that oxygen. So the uptake of water from the plant is not going to stop, right? The splitting of water happens in photosystem 2, right? And photosystem 2 comes before photosystem 1, right? So it's kind of that first step, and we have a mutation in like the third step of the light reactions, right? At first, it's not going to cause much of a difference, right? We can still split apart that water to produce oxygen, right? After a while, though, right, things are going to start to back up, right? If photosystem 1 has stopped working, right, it's essentially no longer going to excite the electron that's been given to it, right? So all those other excited electrons that are coming its way is going to be backed up because they can't be moved through photosystem 1, and eventually we're going to stop producing oxygen, right? So it's kind of a double answer here. So yes, oxygen won't be affected at first, but eventually things will get backed up and it will stop. Does that make sense? All right. All right, this is where I have a built-in break here. And I'll actually say, how much time do we have? Let's take a half hour, right? I have 15 minutes built in to this activity. I want you to draw out the light reactions because in my experience, muscle memory is everything, and having you actually have to figure out how to draw it is a really great study tool. I want you to include all these different things in your drawing, and then you'll turn it into me, right? So I kind of allotted 15 minutes for that, but I'm going to say you'll have 30 minutes because if you want to take a 15-minute break after this, if you want a little bit more time to draw, we have some wiggle room, right? So we will resume lecture at 11:45, right? And in the meantime, I want you to work on that drawing, take a break, whatever you need to do here. Yes?
Can you work in small groups, or do you want it done in
You can work in small groups, but I want everybody to turn theirs individually in. So yes, you can work together to make this. I did put in here to include the cytochrome complex. I'm actually going to delete that because we didn't talk about it. So don't worry about the cytochrome complex.
No. That's all I got.
I actually forgot to grab some colored pencils. I'm going to go grab some, so if you want colored pencils, they have colored.
Oh yeah, true.
I'm pretty sure I have the same notes drawn. Who did someone that the state and say they get a circle answer and lost a quarter of a point, but they have extra credit. But also wrote about Indiana Jones and the other dots. So I feel like I grasped the concept more.
Oh wait, I think this is the same one. Oh, it's fine.
Let's see.
I just
Yeah.
zoomed in on it a little bit.
Don't use this, it's cheating.
You're all
It's
like, I
cheating.
just learned this. I just saw my face. I'm gonna look at it. What? Okay, wait, my question is, I just invited this in spring. Okay, so we're in my notes.
It's literally on
It has the membrane. Wait, the membrane is. Wait, oh, it's the.
Where is it?
I'll probably just draw it.
Look at it.
And feel weird to like skip it. I'll just draw like.
And there's the same complex.
Okay, so this has a membrane. What are these little? These are the
This is the membrane.
So we don't need this here. This is my I feel like.
It's the plasma membrane for the cell.
Okay, I'm gonna start with the phosphorus. That's the bottom of the drawing. Oh, that's very. Okay, I already put three.
Sorry.
The drug little guys.
Little guy.
Yeah. Real fast.
That's a trick question. She put this one. Suddenly, like, yeah.
It's a bad idea to do this with that.
Really?
Colored pencils, markers if you want.
What happened?
There's this one song that I want to sing.
I'm trying to keep it as simple as I can, but I can't put out easier. All right, so we left off with the light reactions, right? Creating those three products, creating oxygen, creating NADPH, and creating some ATP, right? We're going to move into the next step of reactions in photosynthesis here. And we're going to look at the Kelvin cycle, right? Sometimes referred to as the dark reactions because you don't need light for this to happen. Dark reactions are a little misleading though because it implies that it happens in the dark, right? It can happen during the daytime too, which is why we now refer to it as the Kelvin cycle, okay? It's in this Kelvin cycle that we are using carbon dioxide from the air, so CO2 from the air, taking that carbon and incorporating it into sugars that the plant can then utilize, right? So when we look at the Kelvin cycle, we're going to see that this process takes a massive amount of energy, right? Which is why the light reactions are so important, because we created these energy carrying molecules, right? We have ATP, we have NADPH that we can now use to make these sugars, right? Now the product of the Calvin cycle is sugar, right? But directly out of the Calvin cycle, it's not called sugar quite yet because there's a little bit more processing that has to happen. The Calvin cycle is creating this product called glyceraldehyde 3-phosphate, or G3P, right? If you tell me G3P, I would accept that on any exam or assignment, right? I like to remember glyceraldehyde 3-phosphate because there is also an acronym of 3GP, which can get very confusing for G3P, right? But we're producing glyceraldehyde 3-phosphate that is then made into glucose, right, or a sugar. All right, let's dive into this cycle, right? There's a lot happening. We're going to focus on the basics and the different stages that is being incorporated here, right? So if we look at this cycle, right, we can split it into 3 parts, right? We can split it into this first phase, which we would call carbon fixation. We can split it into the second phase here, which is reduction and carbohydrate production, right? The stage where we're actually producing those carbohydrates. And this third phase here, which we would say is the regeneration of RUBP, right? And we'll talk about RUBP and why we need to regenerate it here, right? This is a big picture. We're going to break this down stage by stage, right? We're going to start with this first phase. We're going to start with carbon fixation, right? We're taking that carbon dioxide and incorporating it into other molecules to go through this cycle. Right. I'm trying to slow down. All right, carbon fixation. So we have CO2, right, in the air. It's coming in through the plant, through those stomata. It's interacting with those plant cells. That CO2 is going to be combined with this molecule called RUBP. Right? We're taking that carbon dioxide, we're combining it with another molecule in order to make 12,3-PG. Right? This is where I said that those acronyms, there's a 3, I see even I mess them up, 3PG and G3P, right? Which is why I like to memorize the full name, right? But we're taking carbon dioxide, incorporating it with RUBP to make 3PG, right? So 3 phosphoglycerate molecules, right? Now in the process of this happening, we have an enzyme, right? So an enzyme is helping to make this reaction happen a little bit faster here. We have this enzyme called Rubisco. Rubisco is such an important enzyme, right? It's actually thought to be the most abundant enzyme in the world, right? It's this enzyme, right, that speeds up this reaction of bringing carbon dioxide, combining it with RUBP to create 3PG, right? So that's the first step, right? We're fixing that carbon dioxide into a molecule. In this second step here, we're actually going to make the sugar, right? So in this second step, there's a series of reactions that occurs. Right? We're not going to focus on the reaction. What I want you to focus on is big picture here. Right? We are taking this molecule that CO2 and RUBP created, this 3PG, taking it through a series of steps which uses energy. Right? We're using ATP here. We're using some NADPH here. Right? Energy has to be used. And ultimately, we are creating our glyceraldehyde 3-phosphate, our G3P, which is our precursor to a sugar. Right? And we can see that this G3P goes on to make our glucose. Right, so step 2, right, use a lot of energy. We're using that ATP, we're using that NADPH that we created in those light reactions, right, to change, right, our 3PG into G3P, into glyceraldehyde 3-phosphate, which goes on to make our sugars. Right? So great, we have created sugars, right? The sugars can now be used, right? Making sure everybody has time for their notes here. Yeah?
So what is structurally different between 3PG and G3P? Is it like the organization of the phosphate group?
Yeah, organization of phosphate group, not necessarily number of
phosphate is the same, right?
carbon.
Nitrogen is the same as well.
Now it's been a while since I've had to dive in depth on all these different
Yeah, this
changes.
doesn't really pertain to anything that we have to know. This is just
Yeah,
this
yeah.
might be-
It's the way it's configured, right, leads to a better molecule to make sugars.
So it's probably more double chain phosphate groups then,
Potentially,
just because they break down easier.
potentially, yeah. I'd have to look back in my physiology textbook to know those details. I don't even think those details are mentioned in the Bio 101 textbook. Yeah. All right, so we have created our G3P molecules here that will go on to make sugars. Right? One thing I want to note here is in this whole process, right, we're taking 6 carbon dioxide molecules, combining it with 6 RUBP molecules. We're creating 12 molecules of 3PG, right? Those 12 molecules of 3PG will make 12 molecules of glyceraldehyde 3-phosphate. Only 2 of those are going to go on to make sugars.
This is something that pertains to the lecture. Did you pause the recording while we took a break?
Oh, I don't think so, but thank you for letting me check. I didn't.
Okay, I just was confused because closed captions went away. So.
They did. They go away every time I minimize it, so thank you. Yes. All right, so we're producing 12 G3P molecules. 2 of them are used to make sugars, right? Only 2. Right? The rest of them are going to go on to this next phase, right? So 10 of those molecules will stay in this cycle and will be used in this next phase, right? Called the regeneration of RUBP, right? So remember, RUBP is that molecule that first combined with that carbon dioxide, right? We created it into an entirely different molecule, right? And this second step, we created 12 G3P, glyceraldehyde 3-phosphate. 2 of those were used for sugars. We're left over with 10 of those molecules, right? We now have to make more of this RUBP molecule to keep this cycle going, right? So these 10 glyceraldehyde 3-phosphates are going to be transformed back into 6 RUBP, right? And in the process, using even more energy, right? So we're using energy in this stage as well, right? So the end result of phase 3 is now we have RUBP, right? We have the molecule RUBP, which means we can go right back into phase 1, incorporate more CO2, make more sugars, right? Regenerate RUBP again, incorporate CO2, make more sugars, right? And the cycle just keeps on going, right? That's all there is to the Kelvin cycle. Like I said, I think it's less complex than the light reactions, but note that I've also very much simplified it.
It doesn't pertain to the lecture, but I figured out the difference. So 3PG has an oxygen attached to its double oxygen chain,
Okay.
and the glyceride
Glyceraldehyde 3-phosphate.
has a hydrogen ion. So it's just 2 different ions that are attached. So it's when H2O comes into this, or the CO2, sorry,
Mhm.
CO2 comes into the cycle, it takes that carbon takes the carbon ion away from it to add, so the RUBP can split into two, so it takes one of the carbons and adds it to the RUBP, so there's 3 carbons on either side, and then it takes the oxygen from one and adds it onto the tail end of it, and after that, the oxygen is taken away from the ATP reaction to get the P3P.
Awesome. Great job for looking that up. We can kind of see that happening in these structures here. Yeah. So, like I said, a
Make
lot
it more complicated.
going on. It is actually very complicated. We're sticking to our 3 steps though, right? But know that, yes, we can go further into depth, right? So we have our 3 cycles: carbon fixation, right? Reduction and carbohydrate production, and the regeneration of our molecule of RUBP. Right? Before we leave the dark reactions, I just want to reiterate how important that enzyme Rubisco is. Right? So that enzyme is helping to incorporate the CO2 into RUBP. Right? It has this fun long name here, right? I don't even try to remember this long name. I call it Rubisco. Most of the world calls it Rubisco. In a scientific paper, you might see it as ribulose 1,5-biphosphate carboxylase oxygenase, right? It actually has the ability to also use oxygen instead of just carbon dioxide, right? Which is why some plants have these really cool adaptations and how they do photosynthesis, right? I don't know that we are ever planning on or have taken those alternative ways of photosynthesizing into space. So we're not going to talk about those alternative methods. We're sticking to the basics here, right? But it's found in all photosynthetic organisms, which is why we think it's really the most abundant enzyme on Earth. All right, that was a lot of information again. Let's take a step back and review the Kelvin cycle, right? As a note, I incorporated this very diagram into my last exam in Bio 101. I love this diagram. It might show up in some form on your next exam, right? So I would definitely use this as a study tool here, right? I want you to fill in the blanks, right? What is missing from this diagram? And then we will go over it.
Oh, 3.
Kelvin cycle versus light reaction. You feel better about it?
Yeah, I like it better.
There's fewer things to remember and conceptualize. It's a lot of terms you have to remember. Yeah.
This is like,
Yeah, it's a cycle.
I do too. I just
How's it going?
Oh, so it's, phase 3 is using the G3P that we made in phase 2 to
Oh, Rubik's Cube. That's why it's a
So
second.
it's using another 18.
I'm gonna give you a little bit more time here.
To then transfer and combine the G3P molecules. So it takes one carbon ion out and sticks it together. And then there's a byproduct. It doesn't form that byproduct.
I'm actually gonna give you another couple of minutes because I see we're still not done filling in the blanks on our own notes.
And then that creates
Feeling okay about dark reactions,
I
Calvin
think so.
cycles?
A lot better than I thought.
Yes. I get the hard part out of the way first, right?
Yeah.
Yes.
That's the process.
Only two, yeah.
You could never
For sure.
about if you find out your CC before returns.
Should do. Yeah, should do. Oh, yeah. That would be the hard. Yeah, the 10 of them. 6.
Raina, did you hear they're opening Pippin next year?
Ooh, the super modeling, right?
I don't know. I thought they just closed it because it like costs less to run, like
Yeah,
keep
the one
open.
on the top.
Because carpet compared to tile. That's like
No, you're right,
the
you're right.
difference between
No.
Story.
Yeah, not the one on the left.
But Story is all
You're
gonna
right.
be single rooms now. Like
Interesting.
people that want single rooms.
Oh.
Wait, when?
Next year.
This year? Wait, how, are they gonna break it down and rebuild it again, or how are they gonna?
No,
do
like
singles?
they're just gonna like let one person live in. So like an entire room Story would be a
That
single.
would be nice.
Yeah. It's interesting.
Yeah.
Sorry was my freshman dorm,
Same.
so.
Sorry,
Yeah.
sorry.
All right, let's go over our Kelvin cycle here. Really these blanks, besides Rubisco, right? All these blanks include everything I would expect you to know of the Kelvin cycle, right? In addition to Rubisco. All right, so what is this first blank up here? What are we incorporating into the cycle? CO2, yes. And that makes what molecule? Starts with a number.
3PG.
3PG. Yes, during our first phase of carbon fixation. Yes. Oh, I thought they came up one by one, but they don't, right? Our next phase, all right, would be that reduction and carbohydrate production, right? This box here, right, you're making the G3P, which goes to make our glucose molecule, right? We're then recycling the rest of our G3P in the regeneration of RUBP stage in order to regenerate our RUBP molecule. Are there any questions on the Kelvin cycle?
Is there another molecule that is being released in the reaction of G3P recycled into RUBP? Or is it just a TP to ADP losing a hydrogen ion?
I don't know. I would have to look into it. If there is, it's very minor because it's not really mentioned in like one-on-one kind of senses. I want to say no, but I could be wrong. So I'm not sure.
Probably not, but like there's a chance it's oxygen.
Yeah, there's a chance that something's released. I don't think so. I could be wrong. Yeah. All right. All right. Before we go on to an activity, right? I just want to mention the homework. Right, I'm having you watch a series of videos. I believe they're all pretty short. Annotated bibliography is due tomorrow, so hopefully you've been working on that. You've got some heads up on that assignment. You only need to find 4 sources for that annotated bibliography, so likely you'll end up using more for your final project. The How to Grow Your Planet assignment is due on Friday. Has anybody had a chance to watch it yet? Some of you? I enjoy it. All right, and then exam 2 on Monday here, and that study guide is online. All right, so I had you listen to the Gardeners of the Galaxy podcast for the quiz today, and they talked about how many plants you might need to keep an astronaut alive, right? Let's think about that in terms of campus here, all right? So I have a scenario presented to you in this document. You can either click this link or it is linked up on Moodle under today's date, right? And my scenario is, right, there has been a deadly virus going around, right? Maybe it's another round of COVID, maybe it's something different, maybe it's like, what's that show, The Last of Us? Yeah, with the fungal infections going around. Something has happened. You need to seal yourself in your room, right? And we want to figure out how many plants would you need in that room with you in order to keep you alive, right? For the sake of the argument, we are going to ignore all your other basic necessities here, like food and having to leave to go use the restroom, water, all right? We're going to assume that we're in a completely self-sustaining dorm room, right? And we need to provide enough oxygen. How many plants would that take? So if you go to this document here, all right, we can calculate this out. It was not. I had looked up a picture of an example dorm room for like prospective students to Gustavus, and I put a lot of different plants in it. We can calculate out how many plants we might theoretically need. Now we're making a lot of assumptions here, and that's one of the last things I'll ask you is what assumptions we're making, right? We know that a typical human being breathes out about 420 liters of air per hour, right? We also know that air is about 20% oxygen, so we would need about 84 liters of oxygen an hour to keep us living, all right? Now an average leaf, right, creates about 5 milliliters, or 5 milliliters, or 0.005 liters of oxygen per hour, right? So we need to calculate out how many leaves would we need of average size, right? And when I'm thinking of an average size leaf, that's why I brought in our plants here. I'm thinking typical average houseplant leaf size might be something around like this, right? How many leaves would we need to keep us alive, right? That's the first calculation. Once you figure that out, right, we're going to make another assumption that the average houseplant has about 25 leaves. This one, I counted it out, has 17 leaves, all right? The average houseplant, 25 leaves. How many plants would we need to provide us enough oxygen? Right? And then the last piece of the puzzle that you'll work through is figuring out how many plants you could actually fit into your dorm room. All right? So we're going to say that an average plant needs 1 cubic foot of space. I've converted this into a metric unit for you. What you need to do, and there's a link on the end of this document here, is look up the floor plan for your dorm, right? If you don't want to look up your specific dorm room, you don't have to, but all the floor plans are up on Gustavus here. Find the floor area in centimeters, divide that by the amount of space one plant needs to take up, right? And figure out how many plants you can fit into your dorm. And see if we're anywhere near how many plants you would actually need to keep us alive, right? Now there's a lot of dorm rooms represented here. If you are living off campus, you can choose a dorm room. Maybe you lived in the past to analyze here. And then we will write the results up on the board to see maybe which dorm room you should select if you think there's going to be a deadly virus coming our way. All right, I'll let you work through this. Sure. it's more calculations, but yeah, go for it.